In this video, we will explore the processes of mitosis and meiosis in depth. We'll break down each phase of both cell division methods, prophase, metaphase, anaphase, telophase, and cytokinesis, highlighting their key features. Additionally, we'll examine the fundamental differences between mitosis and meiosis, including their roles in growth, repair, and reproduction, and how they contribute to genetic variation. Let's get started. Understanding mitosis, meiosis and genetic inheritance through modern research. A group presentation by Nishat, Lilianne, Amaima and Ayesha. In doing our research, we came across three very informative articles. The first one was called The Evolution of Meiosis from Mitosis. Written by Adam S. Wilkins and Robin Holliday. Key point from this article: meiosis likely came from mitosis, but it added several complex new steps. Chromosome pairing and swapping DNA. Meiosis is a big mystery in evolution. Scientists like Maynard Smith and Hamilton saw the origin of meiosis as one of the toughest problems in evolution. The next article that took us through each phase of mitosis and meiosis was Germana. Provided by Academic Center for Excellence. Summarizing what we learned in this article is as follows. Mitosis is for growth and repair. Happens in body, somatic, cells. One cell divides once to make two identical daughter cells. Each new cell has the same number of chromosomes, 46 in humans. They are diploid. Goes through these stages: interphase, prophase, metaphase, anaphase, telophase, cytokinesis. No mixing or swapping of genetic material occurs. In the other hand, meiosis is for reproduction. Happens in reproductive, germ, cells like sperm or eggs. One cell divides twice to make four non-identical daughter cells. Each new cell has half the number of chromosomes, 23 in humans. They are haploid. Involves crossing and independent assortment, mixing of genes for genetic diversity. Two major parts. Meiosis I. Chromosomes pair up and exchange segments. Meiosis II. Sister chromatids split like in mitosis. Lastly, we have an article called Mitosis, Meiosis and Inheritance. Written by Ilona Milo, Ph.D. Published in 2008 by Nature Education. Historical insight. Scientists Walter Fleming and Walter Sutton used drawings to study chromosomes during cell division. Walter Sutton's work supported Mendel's principles of inheritance by showing that chromosomes are connected to how traits are passed down. Goal of research. Nature citable. Mitosis, meiosis, and inheritance. Hypothesis, research focus. The article compares mitosis and meiosis, focusing on how each process plays a role in passing on genetic information. It doesn't test a specific idea but explains how these processes lead to both genetic variety and stability. Methods, procedures. Review of existing research. The article pulls together information from different studies and past observations to describe mitosis and meiosis. Visual aids. It includes diagrams of chromosomes during mitosis and meiosis to help show how they move during cell division. Historical context. The article mentions past research and discoveries to give a background on how we understand genetic inheritance today. Methodology. As mentioned in Germana Community College, mitosis versus meiosis. Hypothesis, research focus. The main aim is to explain the roles of mitosis and meiosis in cell division and how genetic traits are passed on. It's not about testing a hypothesis but rather about teaching the differences between the two processes. Methods, procedures. Comparative analysis. It looks closely at mitosis and meiosis, showing how they're different and alike. Clear language. The document uses plain language to explain tricky biology stuff, so it's easier to learn. Diagrams. It has diagrams that show the steps of mitosis and meiosis, which makes it easier to see what's going on. Methodology. As mentioned in Genetics 181, research article. Hypothesis, research focus. The writing is about how chromosomes move when cells split, focusing on mitosis and meiosis. Methods, procedures. Experiments. They watched chromosomes during cell splitting. Checking data. They studied how chromosomes separate to see how traits pass on. Using math. They used math to understand what they saw and figure out what it. Let's put some of this information we've learned together. Let's talk about mitosis and go through each phase. The somatic cell cycle is the process a cell goes through to grow and divide. It has two main parts, interphase and the M phase. 1. Interphase. This is the longest part of the cell cycle. The cell prepares for division. G1 phase, gap 1, the cell grows bigger. It makes proteins and organelles. S phase, synthesis. The cell copies all of its DNA so each new cell will have a full set. G2 phase, gap 2, the cell keeps growing. It checks for any mistakes and gets ready to divide. 2. M phase, mitosis plus cytokinesis. This is when the cell actually divides. Mitosis, nucleus divides, prophase. DNA coils into chromosomes. The nucleus starts to disappear. Metaphase, chromosomes line up in the middle of the cell. Anaphase, chromosomes are pulled apart to opposite ends of the cell. Telophase, 2 new nuclei form around the chromosomes. Cytokinesis, cell splits. The cell divides into 2 new cells. End product, 2 identical cells, each with the same DNA as the original cell. Now let's look at meiosis. Before meiosis. Interphase before meiosis begins. The cell grows and does normal work. DNA is copied during the S phase, so each chromosome has 2 sister chromatids. The cell prepares for division. Meiosis I, reduction division. This is when homologous chromosome pairs are separated. 1. Prophase I, chromosomes coil and become visible. Homologous chromosomes pair up. Crossing over happens, they swap DNA for variety. The nuclear membrane breaks down. Spindle fibers form. 2. Metaphase I, homologous pairs line up in the center of the cell. Spindle fibers attach to each pair. 3. Anaphase I, the pairs are pulled apart. Each side gets 1 chromosome from each pair. Sister chromatids stay together. 4. Telophase I and cytokinesis. 2 new nuclei may form. The cell splits into 2 haploid cells. Each has half the number of chromosomes. Meiosis II, separation of sister chromatids. This happens in both of the 2 cells made in meiosis I. It's like mitosis. 1. Prophase II, chromosomes coil up again. Spindle fibers form. The nuclear membrane breaks down, if it reappeared. 2. Metaphase II, chromosomes line up in the middle of each cell. 3. Anaphase II, sister chromatids are pulled apart to opposite sides. 4. Telophase II and cytokinesis. New nuclei form around the chromosomes. Each cell splits again. End result of meiosis. 4 non-identical haploid cells. Each has half the number of chromosomes as the original cell. These are sex cells, sperm or egg. Discussion. Significance of the results. The Genetics Journal article reveals how meiotic recombination is tightly regulated, ensuring accurate chromosome segregation and preventing disorders like trisomy. The Nature Citable article reinforces that both mitosis and meiosis are central to genetic inheritance, with errors leading to genetic variation or abnormalities. Research connects cellular processes to real-world implications, like fertility, evolution, and genetic disease mechanisms. Discussion. Strengths and limitations. Strengths. The OUP article uses molecular genetics and genome-wide analysis to track crossover sites and recombination accuracy. The citable resource simplifies complex topics for students, making foundational understanding accessible. Limitations. The OUP study is based on model organisms, e.g., yeast, so results might not fully apply to humans. The PDF is descriptive, not research-based, so it doesn't offer new data or findings. Some mechanisms like epigenetic influence on recombination need further study. Discussion. Conclusions and future directions. All sources agree that accurate mitosis and meiosis are essential for genetic stability. The OUP article supports the hypothesis that regulated recombination maintains chromosome balance. Future research should explore epigenetic regulation of recombination, apply findings to human models, investigate links to fertility issues, cancer, and genetic disorders. Thank you. We hope this video was as fun and informative to watch as it was creating it.